At the very outset, I am very much thankful to the organizers of Anusandhan National Research Foundation and IIT Mandi National Webinar Competition 2026. The research presented in this webinar is based on the core research grant project granted by SERB in 2016. The topic of my presentation is liquid crystal-based biosensor for histamine detection. I am Prakash, serving as an associate professor in Department of Physics at Aligarh Muslim University, Aligarh. This is the outline of my presentation. First, I will tell about the liquid crystals. Then I will discuss the biosensors. Then how do they work? And then I will tell about the liquid crystal-based biosensor. Then I will discuss the experimental technique used in this research. Then resultant discussions. And finally, I will conclude the presentation. So what is the liquid crystal? Basically, the liquid crystal is a unique phase of soft matter formed through the self-aggregation of organic molecules. It is basically an intermediate phase between highly ordered crystalline and isotopic liquid phase. Unlike the glass, liquid crystals have long-range orientational order but short range of positional order. They are optically birefringent that results in various polarizing texture and colorful patterns. They have the order parameter ranging from 0.3 to 0.8. So what are biosensors? Basically, the biosensor is described as a tool that quantitatively measures the presence or action of a biological analyte. The purpose of biosensor is to detect substances that are either dangerous or essential for life. The idea for sensor grew out of a breakthrough made by the University of Wisconsin medicine researcher in 2001. In the 20th century, the coal miner used a bird as a warning symbol for buildup of toxic gases. You can see the different biosensors like pulse rate sensor and body temperature sensor, diabetes sensor, and the virus sensor. How biosensor works? An analytical device which is combination of biological component with a physicochemical detector used for the sensing an analyte. The biocatalyst converts the substrate to product which is determined by the transducer. The transducer converts it to an electrical signal and the output from the transducer is amplified, processed, and displayed. So what are liquid crystal based biosensors? Basically, liquid crystals are known to change their ordering and optical appearance in response to the external stimuli such as temperature, electric field, magnetic field, or biomolecule. As a result, they are very useful in everything from cell phone display to the environmental sensor. When the substrate is bonded with the liquid crystal, then the orientation of liquid crystal changes. And the liquid crystal modulates the phase and the polarization of the light that propagates through it. The change of the LC surface on detection of biomolecules is observed by the change in optical signal that can be amplified and processed using polarizing optical microscope, which can be observed through the dielectric studies also. Liquid crystal based biosensor for histamine detection. So basically, histamine poisoning is the most common cause of human illness, and detection of this harmful substance is very crucial. So in this work, we illustrate the relative selective detection of histamine at different concentration varying from 20 milligram per liter to the 500 milligram per liter. The proposed biosensor is based on the orientation transition of a liquid crystal by enzymatic reaction of biomolecules at LC solid interface. The biosensor response is measured through the optical texture of scanning electron microscopy, dielectric measurement with the help of LCR meter. So what is histamine? Basically, histamine is a dibasic vasoactive amine that is present in most body tissues of animals and plant. The concentration of histamine is large enough in the lungs, skin, and gastrointestinal tract. It is stored in mast cell and basophils as a single peptide chain protein that function as a neurotransmitter. Histamine is a substance that is produced by the cell through a peritoneal cavity or connective tissues if there are some antipathetic inflammatory reaction. A large amount of histamine is found in seafood, some salted and dried fermented products. And basically, histamine poisoning affects the human body by binding with the receptor on cellular membrane in different part of the body. You can see that how the different histamine receptor bind with the histamine, and these are the symptoms of histamine on the human head. So basically, in the experimental part, we prepared a self-assembled monolayer of surfactant and silane by dipping the glass slides in the 1% of surfactant and 3% of the silane solution in ethanol at 80 degrees centigrade for 2 hours. Immobilization of diamine oxidase enzyme covalently immobilized to the surfactant and silane film using the glutaraldehyde as a cross-linker. And DAO solution was prepared in phosphate buffer, was sprayed over the same and kept at 30 degrees centigrade for 3 hours. Histamine solution was prepared in the Triton X100. So we prepared cells for the biosensing. First, the reference cell was prepared by coating the top and bottom glass plate with the surfactant silane SEM, and the counter cell or the enzyme cell was prepared. Then finally, a biosensing cell was prepared with the histamine solution, and different concentration of histamine were loaded on the enzyme cell surface and kept at room temperature for 2 hours. And in this slide, you can see the surface morphology on introduction of surfactant, silane, enzyme, and histamine through the self-assembled monolayer or the scanning electron microscope. So you can see the homogeneous morphology of surfactant monolayer in figure A. In figure B, you can see the self-assembly of this surfactant and silane solution. And in figure C, you can see the immobilization of the enzyme, that is DAO. And in figure D, you can see the wave-like structure that is observed after the histamine was introduced on the self-assembled monolayer. In this slide, we can see the surface topography of various concentration of histamine through the scanning electron microscope. We can see the cloudy structure that is formed over the histamine layer, and these grow with the increasing concentration of the histamine. These structures are created due to the enzymatic reaction occurring between diamine oxidase, that is enzyme, and histamine releasing ammonia, hydrogen peroxide, and imidazole for acetaldehyde. This is basic slide that shows the sensing mechanism through the polarizing optical microscope. In the figure A, you can see the reference cell that is in homotropic alignment or the vertical alignment that shows the dark field of view. In figure B, that is the enzyme cell. And in figure C, that is the biosensing cell in which the diamine oxidase was introduced. And in figure D and E, you can see the different concentration of histamine was loaded, and we can see the change in alignment from vertical to the planar, in which we can see the bright texture under the polarizing optical microscope. And we can also confirm this sensing through the dielectric spectroscopic measurement. We can see the dielectric permittivity of the reference cell, which is almost 4.56. That as we increase the concentration of histamine, then this permittivity decreases due to the change in alignment from vertical to the planar. In planar alignment, the permittivity is less because the molecules is perpendicular to the surface. In case of homotropic alignment, the molecules are parallel to the surface. And we can see the value of dielectric permittivity in this case of 8Cb molecule are homotropically aligned in the presence of surfactant. And when enzymes are incorporated in this SEM, then tend to align the homogeneous direction and the value become decreased. So when histamine is incorporated with the concentration on the enzyme coated SEM surface, molecules remains in homogeneous state and there is only slight variation in the dielectric constant. So this research has also been published in Material Letters, Biosensor and Bioelectrics, Resonance, and the technology was also patented as an Indian patent number 548488. And the detection of histamine is done at different concentration, varying from 20 milligram per liter to the 500 milligram per liter. And it induces a definite birefringence by disrupting the molecule at the interface. At the concentration of histamine is increased, we get homogeneous alignment with improved contrast. And the sensing of histamine was achieved through the conversion from homotropic to the planar alignment, which is the basic sensing phenomena in the biosensor. And the dielectric behavior were observed in a wide frequency range from 20 Hz to 2 MHz by a high precision L-cell meter. And when enzyme and histamines are incorporated in SEM, then molecules tend to align in the homogeneous direction. Thank you so much.